When Bob Desimone asked me if I would introduce Corey today, my first thought was that I shouldn't do it. I suspected that it wouldn't be fair to Corey because I would say so much, by the time I was done, there'd be no time for her to give a talk. But I said yes, and I'll try to be quick. So, Corey was born in Virginia, received her BS in biochemistry from the University of Georgia, was a PhD student with Bob Weinberg, and did her postdoc training with me. In 1991, she became a faculty member at UCSF, and then in 2004 moved to be the Torsten Wiesel Professor at Rockefeller University. Okay, so that was quick. But I'm not going to stop. I'll fill in a few of the details at this point. I first got to know Corey, it's amazing, 36 years ago. Corey was a TA in my genetics course. And to be succinct, she was a spectacular TA in every regard. The same year, 1982, she published her first paper in Nature. And in the context of today's event, I think her co-authors are noteworthy because they included not only Bob Weinberg, who was her supervisor, but also Ed Scolnick. Okay. The paper defined the molecular basis of the activation of the proto-oncogene RAS. And RAS had been discovered a few years earlier by Ed Scolnick. And to this day, it's at the core of what's probably the best studied and best understood signal transduction pathway in cancer biology. As a PhD student, Corey made many contributions to the molecular understanding of cancer, including a landmark paper in which he cloned the new oncogene. New is the rodent gene equivalent to what is sometimes called HER2. HER2 is the target of the drug Herceptin. Okay. Herceptin has basically revolutionized the treatment for a particularly aggressive form of breast cancer. Corey joined my lab as a postdoc in 1987. I think I remember her application to the Helen Hay Whitney Foundation, in which she described her experience at a poultry slaughterhouse and explained how that turned her into a vegetarian. Okay, she got the fellowship. Okay. As a postdoc, Corey began studies of the C. elegans chemosensory system, and she initially focused on soluble chemicals, namely taste, but then she transitioned. She guessed that C. elegans has a sense of smell, and to pursue that possibility, she brought into the lab odors that she suspected C. elegans might like. Okay, now to think about this, you have to know that C. elegans eats bacteria that grow on rotting matter, plants and animals. Okay, so the odors that C. elegans might like, I would say most of us in this room would have a different response to, which was also true for her baymate, who was not terribly fond of these experiments. Nonetheless, Corey persevered, and she discovered that C. elegans indeed has a sense of smell, and she has spent much of her career building on that discovery. This morning, I looked up the letter of recommendation I wrote for Corey when she applied for faculty jobs first. And I ended that letter with the following sentence. In short, Corey Bargmann is a truly remarkable young investigator, and I anticipate that she will have a brilliant career as an independent researcher. In short, she is, and she has. As a faculty member at UCSF and Rockefeller, Corey has made a series of landmark discoveries concerning the relationship amongst genes, neurons, neural circuits, and behavior. For example, she demonstrated that a particular 7-transmembrane protein is the molecular receptor for a specific odorant. thereby arguably proving the hypothesis that Linda Buck and Richard Axel had made previously. Richard is here today, and I have to say he must not have wanted Corey as a competitor. Today, he and Corey are husband and wife. Okay. Corey also discovered a molecular mechanism involved in breaking left-right symmetry in nervous system development, and established that this asymmetry is of behavioral consequence. She discovered that a single mutation in a neuropeptide gene determines whether animals are solitary or social feeders, a behavioral difference that later proved to be a reflection of differential responses to the level of environmental oxygen. Corey showed that sensory neurons encode behavioral responses to an odor through pre-wired neural circuits, which neuroscientists often call preference maps or labeled line systems. Corey also showed that olfactory adaptation is a circuit-level phenomenon in which interneurons feed back onto primary sensory neurons to alter their properties. And I could go on and on with the science, but Corey is going to do that in a more succinct way than I could ever do. I also want to say Corey has made exceptional contributions not only through her scientific discoveries, but also through her service to the scientific community. Most notably, she co-chaired with Bill Newsom of Stanford the BRAIN initiative that was announced by President Obama in 2013. This was an effort with many complexities, scientific and political, and I think that Corey navigated the issues adroitly, and I think this effort is now greatly benefiting the neuroscience community. Corey is a member of the advisory council to the director of the NIH, a council in which I had the pleasure to overlap with her briefly. In addition, Corey has served on many other advisory groups, including the MIT Broad Stanley Center, the Simons Foundation, the Cold Spring Harbor Lab, and Genelia Farm of the Howard Hughes Medical Institute. She's also on the scientific review committee of the Jane Coffin Childs Foundation for postdoctoral fellowships. Corey has received many honors for her contributions. Most notably, she received a breakthrough prize in the life sciences the first year this prize was awarded, and interestingly, the same year, her graduate mentor, Bob Weinberg, also received a breakthrough prize. Corey has been the recipient of the Kavli Prize in Neuroscience, the NIH Director's Award, the Ben Franklin Award of the Franklin Institute, and many others. I could still continue, but I've gone on long enough. Please welcome Corey Bargmann.
So I'd really like to thank the McGovern Institute for the honor of receiving the Scolnick Prize. There are a number of reasons, as Bob says, that this is really a very special honor for me. Most importantly, perhaps that I did train at MIT, and to feel that you've kind of come back and made good in the eyes of your mentors is really a significant, really gives you a significant sense of accomplishment, and I'm very happy to do that. And also, you know, there is that link with Ed Scolnick himself. And, you know, you never know who you're going to run into in your scientific career, whether it's the students you were with when you were a first-year graduate student or someone you collaborated with and you just keep running into over and over. You develop a sense of connection to the community and to science as a whole. And finally, it's always good to have an excuse to visit MIT. And it's always great to talk to the scientists here, and I could easily spend 5 one-day trips here, visiting with, you know, having half-hour appointments with the different faculty in biology and brain and cog and related areas. So I'm delighted for that reason as well. Now, some of you will remember that I gave a lecture here almost exactly 1 year ago, and about the work that we're doing in the lab to understand the C. elegans nervous system. I'm not here to listen to myself talk, but in honor of those of you who are sort of hearing me 2 years in a row, I thought I would do something a little different in my talk here from what I usually do, which is usually I'm very focused on whatever the latest data are and what the story is and communicating that. And today, what I'll do instead is I'll try and take a step back and just address this basic question of what it is to understand a complete wiring diagram and how it is what we feel that we've learned from studying the wiring diagram of C. elegans. So I started my postdoc in 1987, as Bob said, and in 1986, John White and his colleagues published this rather remarkable paper in which they described from serial section electron micrographs the complete pattern of neural activity neural connectivity, in the nervous system of C. elegans that links the 302 neurons together. But this is the pointer. And that is rather fetchingly illustrated in this slide here, showing, basically trying to communicate here to you that not only so C. elegans has exactly 302 neurons. Every single one of those neurons has a name. Every single one of those neurons can be recognized in each individual of the species. It has a reliable pattern of gene expression. It has a reliable set of connections and functions in the context of the animal. Now, you would think that with that information and having that information over the past 30 years, although I should say much of that information has really been acquired on top of this wiring diagram since then, we would really understand how the behavior of this animal works. And what has happened is that the connectome, as it's called, of C. elegans, turns out not to be an answer, but rather a question. And the question is, how is it that a set of known connections between a defined set of neurons give rise to the behavior of the animal? Now, back when I was at MIT, some of the people like Marvin Minsky and the sort of artificial intelligence community that was here at the time got their hands on the wiring diagram and were convinced that they would be able to use some pretty simple computations to solve the nervous system of the worm in short order. And while these people were really interesting and they would come to the Horvitz lab and they were fun to talk to, they would tend to go away and then we would never see them again. And the reason is that it's really not that simple to read a wiring diagram. And so what I'm going to try and do today is to express what we think we've learned about this wiring diagram, how we think it works, why we think it was so hard for us to understand at first. I think there's some things where we just have the shape of an answer or a hypothesis about what the answer is, and those things can still be tested, but there's some evidence for that. So I'll just sort of start you with the basic idea and probably the way that we all thought about it for a long time. And that is the following. There are sensory neurons, integrating neurons, and motor neurons. They're sort of about a third of each. And then there's this group of 20 neurons in the pharynx, which have traditionally been ignored, and which I had to paste into this figure here from a paper on the wiring diagram. And I would say that the dominant thinking about this diagram is that it is hierarchical, that it is feed-forward, and that information is flowing smoothly from different kinds of sensory inputs to the behavior of the animal. Now, I think it is that idea has truth to it, but I think it's reevaluating that set of assumptions that has really given us a different view of the C. elegans nervous system that I will try and share with you today, which gives us a sense of it as much more of an integrated unit, showing collective actions and coordination of activity across large numbers of neurons. And then the second thing I really want to bring out, in addition to that idea of coordination, is the idea about how different time scales are represented in the nervous system and how it is communicating information across these different time scales to generate behavior. And I think that was probably the part of the story that we were most wrong about when we first started thinking about the wiring diagram, was that we came to that with a field as neuroscientists in a field where neurophysiology was really the dominant way of thinking about the nervous system and the one from which we had learned the most about how the nervous system worked. And those were really systems where people had thought a lot about things like ion channels and synapses and action potentials, which in the event C. elegans rarely uses. And we're really in these sort of the time scales really of milliseconds of the rapid transmission of information through the system. Now, the problem with that in C. elegans is if you're trying to use millisecond timescales, by the time you've gotten from the front to the back of the nervous system, only a few milliseconds have passed. These are really it's a very shallow nervous system. There aren't that many layers between the neurons. And so what we've come to realize over time is that there are other time scales through which information is flowing through the nervous system in parallel with these rapid forms of communication. And two of the ones that I'll really bring out today are those timescales of circuit states, which we think may be sort of mapping information over slower timescales, and the timescales of neuromodulation, which we think might correspond to something like motivational states or emotional states. We just call them internal states in the worm. And then, in addition, on top of that, I'm going to talk about new work we've done to look at how the nervous system changes over time in the mechanisms of learning and memory. So it's these longer timescales that shape the combination of the anatomically defined wiring diagram and the fast transmission of information that we think provide the intermediate level that helps us to understand how the nervous system works. So where did we start? So where we started were the sensory neurons. And that has really been, again, a dominant idea, not only in C. elegans neuroscience, starting with Marty Chalfie and the touch system, but also in mammalian neuroscience. I just want to honor Hubel and Wiesel, since I'm here in Boston, and since Torsten Wiesel is my next door neighbor. And that really starting from the sensory input allows you to understand what the nervous system is experiencing and therefore what it might be trying to do. And so starting when I was in Bob Horvitz's lab under his excellent mentorship, using tools that had recently been developed by Leon Avery when he was a postdoctoral fellow in the lab, and some methods that another postdoc named Jim Thomas had developed, I started trying to map out, and then later many other groups mapped out, the functions of different neurons within C. elegans. And we got really good answers from sensory neurons, I have to say. So the worm interacts with its environment in large part by detecting chemicals from the environment through its senses of taste and smell. It has olfactory organs in its head called amphids that detect a lot of different stimuli. And you can assign functions to them at a variety of different levels. So for example, this neuron shown here in red is called ASH. And this neuron is responsible for detecting a variety of different noxious stimuli, high osmolarity, acids, various toxins, also noxious touch to the nose. And then it's also very closely allied with a set of responses to those, which are escape behaviors. So the animal will reverse, withdraw from those, and then move off in a new direction. So both at a sensory level and at a behavioral level, there's a very clear set of functions you can ascribe to the sensory neuron. And then here's the other sensory neuron, the one that I'll return to most today. It's an olfactory neuron called AWC. And this neuron detects a variety of different volatile organic compounds. Over time, as Bob mentioned, I learned to focus my research on the more mild and pleasant smelling organic compounds that C. elegans can detect. There's only so many times your colleagues will put up with cadaverine in the lab. And so we eventually use things, we know that this neuron detects things like benzaldehyde, butanone, isoamyl alcohol, and 2,3-pentadione, a set of rather pleasant fruity odors that are also byproducts of bacterial metabolism. And these molecules predict to C. elegans where its bacterial food might be, and it leads to odor chemotaxis as well as other undirected forms of food search. So these are very well-defined properties. We can also define these properties in different dimensions as well. So since I mentioned the dimension of time, in more recent experiments, in collaboration with Saul Cato, a theory student in Larry Abbott's lab, we have, for example, looked at the time frames over which these different neurons can detect different kinds of sensory information. So this is a system identification method that's been widely used in sensory neuroscience. And we can see, for example, from that the AWC neuron is capable of following very rapidly flickering chemical stimuli. And we can map out the time frame of that detection of these different kinds of stimuli. And it turns out to represent a time frame that corresponds quite closely to the time frame of the animal's natural movement in sinusoidal movement. And that exactly this property, as demonstrated by Saul in collaboration with Yifan Xu and Christine Cho in the lab, allows C. elegans to do a particular computation, allows it to measure odor gradients with respect to its own movement in something like an active sensing mechanism, so that it can direct itself in the direction of an odor by matching its own head oscillations to the odor oscillations it experiences, and then its future future movement in a new direction. And the nociceptive neurons, for example, have quite different temporal properties. Those neurons integrate different kinds of noxious stimuli over time and average it out over time, making a decision about whether to reverse or not. So we can really see a specialization here, a precision, and a matching of the neuron to the behavior at the sensory level, which I think fundamentally misled us for a number of years. Which is to say, it's not wrong. It's just a property that corresponds to what sensory neurons are doing, which is accurately representing information in the environment that is relevant to the animal's behavior. And we can continue that theme of accuracy here, looking at how the neurons respond to different stimuli. For example, if we give a single animal a single odor 80 times in a row, once every minute for an hour and a half, we see that every single time that sensory neuron responds to the odor and gives a nice, reliable response. So the sensory neurons are reliable as well behaved. And these heat maps here, they're just rasters of individual responses. And here the hot response shows, in fact, an activation of this neuron when the odor is removed, which is illustrated here. All very orderly, all very sensible. And you can also link this to what the wiring diagram predicts. So this particular olfactory neuron not only regulates sinusoidal movement, but regulates reversals. In particular, it drives reversals when the odor is removed, because the animal doesn't like it when an attractive odor is removed, and it is signaling its disapproval by doing a reorientation and trying to move in a better direction. And we can follow the C. elegans wiring diagram, and this at some level is going to be the diagram I'm going to show you over and over again, representing the way that we had thought of this wiring diagram, the way that most people think of this wiring diagram. First of all, we can't even cope with 302. That's too many neurons for us. So as you can tell, we've cut it down to, a dozen or so cell types. And then again, we think of it as, we draw it here as feed forward, as hierarchical. The sensory neuron connects to a number of neurons, which also get input from a lot of other sensory neurons. And we and everyone else had been thinking of these as sensory integration neurons, primarily involved in maybe collecting and comparing information from a variety of sensory neurons. Then there were a group of neurons sort of in the middle, slightly undefined functions. And then these connect to a set of neurons that are directly involved in controlling behavior. In this case, this neuron is called the backward command neuron because it is active in association with and can drive reversal behaviors with very high accuracy. So it's really not that hard to get from this sensory neuron to a reversal. You can get there by a direct route. You can get there by a number of indirect routes. It all looks quite logical. Now I'm just going to highlight here 3 of the neurons along the way, because although this way is the most direct route here, this way through this green neuron here called RAM is actually the strongest route, the one that involves the largest number of synapses, which we use as something like a surrogate of what might be the strength of the connection in this system. And so we sort of, with all of this good information about the sensory neurons about which we knew a lot, we turned to these kinds of downstream circuits and asked how the sensory neuron might interact with these different outputs. And here we got a very different kind of result. And this result is that the inner neurons, when we went from the sensory neuron through the 3 different inner neurons that resulted in behavior, the responses of those neurons were actually quite variable. So each of these neurons here is responding to odor, shown in gray. Each of them is shifting from a hot color in red to a cold color in blue. But as you can see, that doesn't happen all of the time. There's actually a fairly substantial fraction of times that doesn't happen, or it happens with a delay. And this variability actually turns out to correspond pretty closely with a variability in the behavior. So we have a sensory neuron that responds every time, and then behaviors that we see sometimes and not others. And so there was something that was slipping between these 2 points that we found hard to explain. Now, our understanding of that came from sort of just going into the nervous system and trying to observe different neurons in detail and recognizing that when we observed these 3 neurons simultaneously, they responded variably, but they responded in a very coherent and correlated way, which is to say, almost all of the time, if one of the 3 neurons responded, all 3 of them responded. And in fact, their activity was time-locked to each other, so that they would then switch on and off in this kind of organized pattern. And this concept of correlated sort of all or none responses, which you also see here, and that the response went from a pretty high and somewhat variable level of activity to really crashing to the floor, turned out to be sort of a useful way for us to recognize that these neurons are not acting as sort of individual processors of information that are adding up information from each synapse independently, but they're acting as some sort of a collective unit. And that collective unit has an interesting property, which is that these neurons can respond either to odor or to behavioral state of the animal. These neurons at every step along this pathway here is showing the response to odor. Again, this is really variable. There's these big old error bars here. It's the sort of thing that makes you think you're technically incompetent. But in fact, what these represent are a set of neurons that responded perfectly well and a set of neurons that didn't respond at all. And here, we have the same neurons actually responding, where instead of just looking at the response to odor, we look at what the animal did in response to the odor stimulus, in this case, odor removal. Here, if the animal is reversed, all three of these neurons became active. And now all of a sudden, there are much smaller error bars, much larger responses, and all three of them are responding together. So this suggests that these neurons are actually responding not to one input just from the top, but from another from the bottom. Can we test that directly? The simplest experiment that indicates that this is the right way to think about this is to use optogenetics to activate neurons at the bottom of this pathway here to activate the backward command neuron and look at what happens to information in the nervous system. And if you activate the backward command neuron, you activate that neuron and you activate these two supposedly upstream neurons. You activate a neuron which has always been described as a sensory integration neuron and drawn as part of the sensory apparatus. In fact, embarrassingly, you activate it more efficiently than an olfactory neuron can activate it. So what this indicates is that information is really flowing bidirectionally, both from sensory information to output and from the motor circuit back up. There's a bidirectional sense of information. And the second recognition from this, which was the work of Andrew Gordis, a postdoc in the lab, is that these two forms of information are actually competing to control the same set of neurons. That the olfactory information has access to it, but is actually fighting against this opposing information, largely from this guy here, who's just a troublemaker, that keeps the animal moving in a different state depending on the ongoing state of activity of the animal. So here's the first sort of intermediate level of conclusion that I want to say about this work, is that it makes this set of experiments has made us really rethink the wiring diagram, that instead of thinking about it as something that moves from here to here, we suddenly have to think about some of these things in between as being interlocked and interrelated to each other. And we have to think about sensory circuits, like the ones that we've been studying, as actually also being motor circuits. And I would say that in just the past couple of years, this idea has also emerged from studies of other nervous systems from very different kinds of experiments. For example, my colleague Gabby Maimon, when he was a postdoc with Mike Dickinson, showed that neurons in the flies that had been characterized for decades as being responsible for optic flow, as being part of the visual system, are highly responsive to whether the animal is flying or at rest. And Chris Neal in Mike Stryker's lab, studying mouse visual cortex, showed that neurons in the visual cortex that showed classical orientation tuning curves, classically visually tuned neurons, actually showed hugely different patterns of activity, again, whether the mouse was resting or running. And subsequent work from Tobias Bonhoeffer's lab showed that if you put the mice in the dark, these neurons respond purely to the animal's locomotion state. They act as motor neurons. In other words, this is not just a question of gain control of visual stimuli. You can strip visual stimuli away from the visual cortex and turn it effectively into a motor cortex. So I think this idea of coherence and feedback existing in nervous systems, even in what you might think of as sensory systems, exists here. And I could have pulled up other examples as well of other sorts of cognitive information, like decision-related activity or reward-related activity, appearing as early as the vestibular nucleus, one synapse away from the inner ear. So I think this could be a principle to think about of coherence. Now, really broadening this is work that has been done independently in Manuel Zimmer's lab in Vienna, who have been looking at whole brain calcium imaging of C. elegans using genetically encoded calcium indicators, which we also use, and in this case, monitoring spontaneous activity. And down in here, you can see neurons flashing on and off, which actually represent the neurons that I was showing you there, AIB, RAM, and ABA. And you can see that there's a correlation in their activity that's not absolute. You can also see other kinds of anti-correlated activity. The point of this is actually that, in general, activity seems to go in patterns here, groups of neurons becoming active or not at different times. And Manuel's lab has characterized this extensively, and more recently there have also been studies from Arabi Samuel's lab across town and Andy Leifer's lab at Princeton. And everyone has basically gotten the same result, which is that even in a nervous system with 302 neurons, those neurons are largely functioning not independently of one another, but actually in collective states, in collective activity states that recruit many neurons into high activity states at a common time. And that can be seen here, again, in this heat map. Each row here is a neuron. Each row is a neuron, that's right. And then this represents the flow of time over about a 9-minute video recording. And what you can see is that there are clusters of neurons that are active together. And these include some of the neurons that I've been telling you about here. And then there are other clusters of neurons that are, for example, anti-correlated with those neurons. So there are multiple patterns of activity moving through the system at a time. And we think this may be a basic property of how this nervous system functions. And putting together this rather extensive analysis that encompasses much more of the nervous system with the work that I told you about from Andrew, we think that the sensory system is, while it is itself a very accurate way of gathering information about the world, has a limited ability to drive behavior. And that what matters in terms of whether a sensory input gives rise to a behavior is the state of ongoing activity in a circuit when the sensory information arrives. That there is ongoing activity, groups of neurons that are simultaneously active, whose state of activity can be permissive or non-permissive for the arrival of that sensory information. And I think that probably is a reasonable perspective to think on many forms of sensory information. There are certain kinds of sensory information that are life-threatening, that require an immediate response. But in fact, most forms of sensory information are relevant under some circumstances and irrelevant under others, and will have different probabilities of driving a behavior, depending on important things like the animal's state of stress, the animal's state of hunger, and so forth. So we think that this information may be more communal, and that this is a different way of viewing what the sensory neurons are trying to do, and again, what the rest of the nervous system is trying to do. And I think that this principle also can be recognized in other very different systems at different levels of complexity. So for example, to move all the way to the completely different level of complexity, which is the human brain, studies of human brain activity, ongoing brain activity independent of sensory stimuli, have indicated that the patterns of blood flow have sort of large-scale, coordinated, slow activity that identify different kinds of networks within the brain. So these networks have names like the default network, or the dorsal attention network, or the visual network, where different regions of the brain will receive apparently higher levels of blood flow on some sort of a timing that appears independent of the ongoing rapid sensory information that a human is perceiving. So here is the time frame of such blood flow signals within the human visual cortex. You can see that it's taking place over a time frame of 10 seconds or so, an extraordinarily slow time by the time frame, again, of channels and synapses. And here's an experiment from David Heger's lab at NYU, looking at the ability of humans to detect very weak visual stimuli at the threshold of detectability, relative to the amplitude of this blood flow signal in the visual cortex. And what you can see is this spontaneous signal dictates how accurately or how well the human is going to be able to detect this signal with tremendous accuracy. And so what this suggests is that, very similarly to what we see in the worm, that really the state of the circuit when the sensory information arrives has a major influence on whether that sensory information is assimilated into the nervous system and into the behavior and cognition, in this case, of the individual. So So what I've told you is that there are circuits, that they have different states of activity. These states of activity, we believe, are developing over time frames of seconds and tens of seconds from the measurements that we and others have made looking at both free-moving animals and restrained animals, and that these sort of second-long times of activity represent such a gate or a preference for different kinds of sensory information coming in and modifying behavior. Now, one of the ways of thinking about this is that what I've told you is that this is a variable, that these circuits are variable, that whether or not you get a behavior out of this is variable or unreliable. And there are different ways of thinking about variability and the roles of variability in behavior. There are actually good reasons that generating variability in behavior is a good way to solve a problem in an environment that is somewhat noisy and unpredictable, or where the value of a stimulus is not itself predictable. But there are other good ways of thinking about variability as well. And one of them is that variability is a really important substrate for learning, that by its nature, if the nervous system is going to do something different, it's going to have to generate some kind of variability to allow it to do something different. And that's most striking in trial and error learning. Trial and error learning implies that you have to try things, that you have to generate variability from which you then select a reasonable outcome. So the best learning circuit, I think, where this idea of variability and learning has been represented is in the songbird and song learning. Your colleague Michael Fee has been one of the important people in characterizing these circuits. And what I'd like to tell you now is that this circuit in C. elegans that I've been describing as a sensory motor circuit, incorporating both sensory and motor information, is also a learning circuit, that embedded within this system are mechanisms that allow the behavior to change over time. So the learning behavior, first of all, in order, C. elegans is not a complex organism. There are things it's going to learn and things that it's not going to learn. And the person who figured out how to teach C. elegans the best in the olfactory system was Yun Zhang, who is now a professor at Harvard in the OEB department, who realized about 10 years ago that C. elegans has an environment that includes both healthful bacteria that are nutritious to the animal and pathogenic bacteria that can kill the animal, and that really this might represent an important thing for the animal to learn for it to survive. And I should say that Dennis Kim, who's here at MIT, was one of the first people to really study pathogenesis and the innate immune system of C. elegans as a mechanism of understanding its physiology. That's been a very rich body of work, and the recognition of that idea that pathogenesis was important in the life cycle was an important trigger for us to study the idea that pathogens would also be important learning signals for the animal. So many of you will know that conditioned taste aversion is one of the strongest forms of learning in any animal. Eating something that makes you sick will lead to a strong aversion to that substance in the future. And what Yun Zhang showed initially in his as a postdoc and has developed extensively since then, is that training an animal, exposing an animal to a pathogenic bacteria, even if you clear the infection and the animal is perfectly healthy afterwards, will lead to a memory and an avoidance of the odor of that bacteria. So Xin Jin, a current graduate student in the lab, returned to that paradigm recently to wonder what kinds of learning C. elegans could show and how long those could last. Now, the original form of learning that Yun had characterized lasts between 12 and 24 hours, which is a pretty good amount of time for an animal that only lives for 3 weeks. But Xin thought about the fact that in many animals, early learning is particularly strong and long-lasting and leads to the strongest memories. And so she reevaluated the learning paradigm that Yun had developed to try to use this kind of early learning and see if C. elegans had that quality as well. And she found that indeed that is the case. So that if she took animals and immediately after hatching exposed them to this pathogenic bacteria, and then cleared the bacteria with antibiotics, coddled them, let them grow up in nice, healthful bacteria until they were adults, and then proposed the same choice to them, these adults maintained the aversive memory of the bacteria they experienced immediately after hatching. We're calling this bacteria imprinting, imprinted memory, just to sort of reflect the idea of behavioral imprinting as something that happens very early in life, forms a long-lasting memory, and is potentially ethologically or ecologically important to the animal. We think this is a real problem that C. elegans has to solve. And importantly, the magnitude of this change after training which is shown here. So what we tend to do, we're kind of on a kick where we want to show like all the data. So this is like every single assay done over a several-month period of naive animals and of imprinted animals. The bars actually represent the mean and something close to the standard error by the time you're done. But I'll be showing you these big wide bars, and actually the effect is quite strong. And in fact, the effect of this behavior, it's as strong a memory in the adult as in animals immediately after training as adults. So this is really a very robust form of memory by the standards of this animal.
Are the number of cells changing?
The number of cells are changing. About 80 neurons get added between the L1 and the adult. Most of those are in the motor system and seem to be just about mobilizing a larger animal. And to the best of our knowledge, they're not critically involved, but we haven't tested them all yet either. And so this here is just to show that this, one of the other criteria that makes us think that this is imprinting-like is that there is a particular time at which the animal can learn this and not other times. So if we train them as first stage larvae, they remember as adults, that's a positive number. But if we train them as second stage larvae or any stage in between, they forget. And so by adults, that response has gone all the way back down to baseline. And here are adults again immediately after training. So there seems to be some sort of a critical period, a specialized period in which learning can occur. And so then the other thing about this behavior is that it appears to be associative. It appears to be selective for the bacteria that they experience. They don't avoid any toxic bacteria. So for example, if we train them on 2 different toxic bacteria here, again, let them recover, let them grow up, and then test them as adults, they only report this learning to the bacteria of training and actually show either no response or even a counter-opposite response to the second bacteria. So it seems to have a selective time of action and an associative response that looks like it's an informative form of learning for the animal. So how does this work? Yun Zhang's lab has gone to a lot of effort to map out the learning circuits for adult pathogen learning in C. elegans. And Xin was really lucky in that she was able to coast along on a lot of those studies that Yun's lab had done, including an important one published in Neuron a few years ago, to identify sensory neurons and interneurons in the circuit that were required for adult learning. What Xin found when she looked at these circuits is that the circuits for imprinting in the L1 overlapped partly but not completely with those required for adult learning. So even though there were differences in the duration of the memory, there was a lot of overlap at the level of neural circuits, sensory inputs, and processing information for these 2 different kinds of memory. There are some differences between them that are informative and important. For example, there's a greater reliance on certain neurons that are much more important for imprinting than they are for adult memory. But one of the conclusions is that these things were not as different as we might have thought. When are the different neurons required? One of the nice things about having a memory that forms in the first stage and performance, a choice assay that you can do as the adult, is that you can try and separate out what's required for learning, what's required for maintaining the memory, and what's required for performance. And Xin silenced neurons at different times in development using a chemical genetic strategy based on a histamine-gated chloride channel that we can use to silence neurons using a transmitter that C. elegans does not use. And then mapped out when these different neurons were required. Were they required during training or were they required during choice? And what she found is that there are different neurons that are selectively required. The neurons downstream of the sensory neurons were either required during learning or required during retrieval, and not during both. There was a clean separation of the neurons within this function. So one of these neurons here is the neuron that we've been thinking about quite a bit as the neuron that generates variability in olfactory responses. That neuron here is RAM that seems to be required during learning. And so Xin dove in a little more deeply here to try to understand what RAM was doing. And one of the things she did was to examine the neurotransmitter that is made by RAM, I should say one of the neurotransmitters made by RAM, which is a biogenic amine called tyramine. And it turns out that animals lacking tyramine, or lacking tyramine specifically in RAM and not in any other location, are defective in learning. So this neurotransmitter here appears to be a key part of the signal that is required for learning in these animals. And you can restore this response to these animals simply by supplying exogenous tyramine. but only if you do it during the time of learning, during imprinting, not if you do it later in growth or during the assay. So this seems to be a signal that is both sort of temporally and spatially restricted to the time of the learning. Now, knowing what that signal is for learning, she then just asked where it was detected. She identified a number of receptors required for learning, but only one of them was specifically required for imprinting, for the formation of a very long-lasting memory. And that one receptor, which is a G protein coupled receptor, is required in one of the neurons required for memory retrieval. So this molecular pathway bridges memory formation on the one hand and memory retrieval on the other hand through a neurotransmitter receptor interaction. So what can we say about this? When we look at this pathway, it actually reminds us of learning pathways in a number of different organisms, that different kinds of physiological learning often involve a separation of functions in memory formation and memory retrieval. And two examples would include the Drosophila mushroom body, for example, where certain neurons that make a modulator, dopamine, are required for the formation of the memory, but not for the retrieval of the memory later. Dopamine is a biogenic amine, as is tyramine. Perhaps there's some similarity there about the roles of these as teaching or learning signals. And then another example goes back to the songbird, where distinct areas are required to learn the song, but not for the retrieval or performance of the adult song. And interestingly, in the songbird, these areas that are required for learning are also areas that generate variability in the song response. And so we think that there might be some sort of a relationship here between this idea of variability and flexibility in generating different learned behaviors. We're currently very excited about trying to understand more about how this learning happens. Part of that just involves returning to the circuits and understanding how they respond to bacteria and how they respond after learning. So for example, one of the things that we've done is to look at the sensory neurons, not just one sensory neuron at a time, but the whole group of sensory neurons in the animal's nose as they respond to different kinds of bacteria. And what we find when we do this is that these sensory neurons actually, it's not just one or two sensory neurons that respond. It's almost the entire group of sensory neurons are aware of the appearance of these different bacterial species. So here, the dark side, there's a cluster of neurons here that come on, and then here in the gray, a cluster of neurons here. Again, dark, these neurons come on, this swath of neurons here, lighter gray, these neurons here. So we're sort of seeing that this really looks like some sort of a population of neural activity is generating a signal about these different bacterial stimuli. Now, this is an alternation between two bacterial stimuli. It turns out that this is also different from giving them one at a time, which has some interesting implications for coding. And we could follow that information in part through the circuit. So for example, Shin has identified one neuron that tracks very closely to whether the animal is showing a memory or not showing a memory of imprinting. That's this neuron RIA. It's one of the memory retrieval neurons, and it actually changes its sign of response to the pathogenic bacteria after the training from a positive to a negative response. Now, we're happy about this, but at some level we know that it's not a complete description of what's going on, because to some level we know that to understand this, we're really going to have to return to this whole network of neurons that I told you about earlier and look at the groups of them that are active at different times. In fact, one of the things that Shin found is that while this neuron changes after learning, so do these other neurons, actually. They change in different ways, sometimes more subtle. We can separate them from the memory. But it's clear that we're going to need to look at this as a whole and not just one neuron at a time to understand the process of learning and memory. But the conclusion of this part of the talk is really that I told you about a sensory circuit that then became a sensory motor circuit, and now it's yet a third circuit. It's a learning circuit. With memory, learning functions, and retrieval functions embedded within the same group of neurons that are involved in generating the basal behavior. In other words, that each of the circuits is really many circuits. The last thing I want to tell you about these C. elegans behaviors is another way in which behavior can change over time. Tyramine, in a way, is a really good, the learning signal that I told you about is actually a good transition to this. When we study the nervous system of C. elegans or many other animals, we really focus on fast transmission of information. But as we study problems like learning, or as we study different kinds of emotional or motivation states, we also discover that there are other ways in which information is transmitted across the nervous system. And those ways often involve other classes of molecules and other classes of receptors for those molecules superimposed upon the classical electrical activity of the nervous system. In humans, these modulatory systems are the targets for most drugs of abuse and most psychiatric and neurological drugs. So we know that they are important for our own behavioral perception as well as those of animals. And they include things like dopamine, serotonin, orexin, and opioids. So these molecules are prominent in all nervous systems. They may actually be a little more ancient to the nervous system than fast transmission, actually based on evolutionary history of some of these things. So they're present in nervous systems across evolution. And in some cases, they may even have functions that are related across evolution. One of the most famous of the modulators, for example, is oxytocin and the related peptide vasopressin, which are molecules known for their effects on social behaviors, mating behaviors, aggressive and mother-pup bonding behaviors of mammals, as sort of symbolized here by the relationship, in this case, of a mother and father vole with their many pups. Now, this is one of the G protein coupled receptor systems. It's important in mammals. It's clearly important in a variety of different reproductive behaviors. And in C. elegans, there is an ortholog of that peptide. There are orthologs of 2 of its receptors. And these molecules have a selective function in organizing reproductive behaviors in C. elegans. They're important for the timing and the coordination of different male mating behaviors. And so without going into a lot of detail about this, what this suggests is that there are important roles of behavioral neuropeptides in mediating what you might think of as long-lasting internal states. That things like reproductive behaviors are special behaviors. They're privileged, and they're represented not just by anatomy, but actually by neurochemistry that helps to rewire neural circuits for their functions. And that, in fact, this particular one appears to be used in reproductive behaviors across all of the major branches of bilaterian animals. Whether that's a conserved or a conversion function, I think we have to take on the soft side. But certainly the idea that this kind of a system, these G protein coupled systems, are being used for slow modulation and for longer lasting behavioral states is a powerful and useful one. So G protein coupled receptors can operate on a time scale of seconds or minutes or hours. We studied some of these when Steve Flavell was a postdoc in the laboratory, and he's now your colleague here at MIT. And he was looking at spontaneous internal states generated by the animal that could last on the order of several minutes, first described by Manabe Fujiwara, that are called roaming and dwelling, that just represent spontaneous locomotion states, apparently unrelated to external sensory stimuli in a one-for-one way, but rather just representing the animal's organized response to its environment. So there's an active state called a roaming state, which involves a high degree of speed and a low rate of reversal, and a quiet state called the dwelling state, which represents a high level of reversal and a low rate of speed. When Steve was in the lab, he asked what neural systems controlled this long-lasting form of behavior, and he identified 2 neuromodulators that have really important roles in this behavior and reciprocal roles. One of them, serotonin, is another one of these classical bioamines that we're all familiar with, and it appears to be involved in lengthening dwelling states. It promotes these sort of quieter, relaxed behaviors. Whereas the second one is a neuropeptide, PDF, and it has the opposite effect. It seems to be required to lengthen roaming states, the more active behaviors. So Steve very carefully mapped out the roles of these modulators by identifying the cells that made each modulator and the cells that responded to each modulator, that made the receptor for each modulator, and which of those were required for each of these different behaviors. And what he found were a variety of cells that make the neuromodulators of different sorts, and a variety of cells that express their receptors. And the important thing to say about these modulators is that the connections that he discovered here were literally completely independent of the classical wiring diagram of C. elegans. They had nothing to do with neurons that were anatomically connected by synapses. So this neuron here is an element of the pharyngeal nervous system, the one I told you that everybody just kind of leaves off their diagrams. This is a motor neuron in the middle of the body. And yet they are coming back and feeding on the neurons within the wiring diagram, including sensory neurons as well as interneurons that were involved in those foraging behaviors I told you about earlier. So this is a completely different logic laid on top of the logic of the olfactory neurons. And so this is another example, really, in which each circuit is many circuits, because we can take that knowledge about neuromodulators and realize that they're being made completely outside the circuit, but they are remodeling the roles of the different neurons within the circuit, and they are actually changing the way that information flows to lead to very large-scale coherent changes in behavioral state. This idea of large-scale and longer-lasting changes in behavioral state has really become very alluring to us. We've been trying to think about behavior now over timescales ranging to the life of the animal. And I'll just close on an experiment that we think is kind of hilarious. In the lab, this is known as the black box experiment, because Shai Stern, a postdoc in the lab, has made a black box, which is literally black, and it is temperature and environmental control and controlled from vibration. And it enables him to monitor the behavior of animals over their entire lifetime, and to try to look at the behavior of individual animals at different developmental stages, compare them to each other, compare different genotypes, compare different conditions, and so on. This takes so much computer time that Rockefeller IT came over to my lab thinking that we were running spam bots out of the lab. I'm very proud of this. And here's just an example of a very simplified summary of some of the data from this kind of an experiment, showing the speed, the average ground speed of animals over a 60-hour period. When they're little, they move slowly. When they're big, they move faster. But here's just something. This speed here represents essentially something like a roaming state. But this is a roaming state. It's actually a constellation of roaming states. That for about this 5-hour period here, the animals were roaming almost all the time. This animal, I should say, was roaming almost all the time. And so we can see much longer-term patterns in this individual's behavior, even than we were able to see before. And one of the things that we've done here is to just sort of start to recognize that there are, in fact, patterns that you can see over the behavior of the entire lifetime of the animal. So here we've normalized the speed on the heat map, so it isn't overwhelming by the fact that the animal is small, and just looked over its entire lifetime. And what you can see is that, for example, in each stage of the larva, it's more active early in the stage than later. And then in the adult, there's a little brief burst of activity immediately after a molt, then a quieter period, and then a more active period that we can only see by looking at very many animals at once. And just in terms of thinking about these longer-term kinds of behavior, here too, we're starting to ask what the rules are that determine how behavior occurs at different times. And the first systems we looked at were the modulatory systems, and I think this would be a fruitful avenue to think about different age-specific, stage-specific, or environmental-specific organizations of very long-lasting behaviors, in this case over days. So for example, this is a secreted peptide, and these animals seem pretty normal when they're young, but they become very quiet later in life. Here are the serotonin guys. Serotonin guys are faster, which is what the red color means, because they're roaming more at all stages in life. But you can see that they preserve the same structure of behavior over time. They aren't affecting this very long-term structure of behavior. They're affecting shorter-term structures within that. And then whereas this other neuromodulator here, for example, seems to have more of an effect on behavioral structure, as well as affecting behavioral state. So we think that these will serve as ways for us of examining behavior over larger scales. And this is my summary of how we're thinking about the nervous system right now. We think that the nervous system, the wiring diagram of C. elegans, represents not a set of instructions, but a set of options. That there are many connections between these neurons, that these connections can vary in their strength or their dynamics based on a variety of different stimuli. Sensory input into the circuit will tend to be accurate, although it is itself subject to some forms of regulation. And then the composition and dynamics within the circuit, which are really being made collectively by neurons within the circuit, are going to determine the outcome and the behavior of the animal. This yellow cloud here in the back is meant to indicate not only that these are acting together, but also that information comes into the circuits from other sources, from sources like neuromodulators, which can reshape these circuits in ways that we still are interested in understanding and that Steve himself will be elucidating in his laboratory here. And really, the final conclusion is that what this represents is that this highly interconnected, highly flexible nervous system is really a single circuit that has many circuits, and that our own highly interconnected, highly flexible nervous systems are also very likely to represent not one set of deterministic circuits, but many circuits which can be deployed in different ways under different circumstances. And with that, I will show you some of the people whose work I talked about today. The work on variability was the work of Andrew Gordis. The work on learning was the work of Xin Jin. And here is Steve Flavel here at MIT, who really established the work on neuromodulators in the lab. Thank you.